I want to start off my talk today with an image that I hope is familiar to many of you, especially here in Maine, where we're lucky enough to not have that much light pollution, especially here in Boothbay, of the night sky. And I'm going to ask for some audience participation, and hopefully many of you have the time and space in your life to enjoy this view now, and if not now, then at some point in your childhood. I think many of us... Is this better? Yeah. Perfect. Sorry. Think about the night sky while I'm doing this. Okay, is this better? Perfect. Okay. So I would love some audience participation in maybe throwing out some ideas of things that you've thought about, or maybe questions you've asked now or in the past when looking at this view. Give me some answers, guys. Is anybody out there? Yeah. What are the stars made of?
It's not exactly a question, more of we are out there. We're part of this cosmos.
We're part of those cosmos. That this is a planet that kind of makes some of us feel small, right? Yeah. Anything else? How do you navigate from that? I use Google Maps. I do not know how to use the stars. And so these are some of these questions. You know, where do we come from? Are we alone? What would it mean if we were to discover life elsewhere? And what I'm trying to get at here is that these are questions that humans have been asking for quite some time. Different cultures, different societies. These are the shared questions of humanity. These are also questions that the scientific field of astrobiology concerns itself with, and that's what I'm going to be talking about today, is this field of astrobiology that thinks about these questions. Where do we come from? Are we alone? So by show of hands, who here has heard of astrobiology? That's a lot of us. Great. Okay. So let me break down the word. The word astro, stars, right? Biology, the study of life. To date, the only place in our solar system, in our universe, that we know out there that has life, that has biology, is this planet Earth. And so what that means is the study of astrobiology really is a story of us right now. This is understanding our origins. How did we get here? How did life evolve and adapt on our planet? And it's really studying life on Earth to help better inform the search for life elsewhere in our universe, how and where we're going to look. This is a extremely multidisciplinary science. It draws on the expertise of geologists and astronomers and physicists and chemists, and then, of course, biologists. I myself am a microbiologist, so I think microorganisms are really cool. And by the end of this talk, I'm pretty sure I'm going to have you convinced, too, that they're about the best organisms we have on Earth. But the focus of my talk today is going to be making the link between understanding life on Earth, and I'm going to be focusing on microbial life, and how we search for life elsewhere in our universe. And to do this, I want to focus on this idea of habitability. So habitability is trying to constrain what places or what environments we think can host life. And we do this based on a number of different environmental factors. So we can think about things like temperature, right? What's the hottest temperature we know life can exist on Earth? Maybe we can theorize about hot places elsewhere, the coldest temperature. Things like food sources, energy sources. Water is another really big one, right? Who here has heard of follow the water? Right? All life that we know on Earth uses water, even if it's in some minute amount, to survive. That's all of the life that we know of so far. And so when we're considering habitability of other places, we're taking into account all of these factors that are based on what we know about life on Earth. So for the next few minutes, I'm going to draw on some examples within our own solar system of some places that we think are pretty interesting in terms of habitability. And of course, Mars. This is the planet next door. This is the one that is in the news very frequently. Mars is an extremely cold and dry planet presently, but in the past it wasn't like that. So about 3 billion years ago, early Mars was a little bit warmer, it was a little bit wetter, it was a lot more Earth-like than it was now. We have evidence that there was large amounts of water on the surface of Mars in the past. We see evidence of old riverbeds that are now dried up. On Mars, we also see the presence of frozen ground, the top of which is very dry. Oh, this is really hard to see. The top of which is really dry, and then below that, ice-rich, hard cemented ground. This is really similar to permafrost that we see here on Earth in the Arctic or in the Antarctic, and I'm going to talk about one of those environments later in my talk. Of course, we've sent some rovers, and we're going to be sending another rover in 2020, called the Mars 2020 mission, that's going to drill into this permafrost. And these are based on what we know about life on Earth. We know that microorganisms can live in permafrost. For example, permafrost microorganisms are something that we hear about in the news a lot in terms of climate change. So we know there are active microorganisms on the Earth, and so perhaps there could be some there on Mars. One of the more interesting places to me in Mars are some of these areas where we think that there could be liquid water. So not frozen water in the form of ice, but liquid water. And this may be difficult to see. This is a time-lapse photograph, some time-lapse photographs where I'll describe it for those who can't see. There's some dark streaks that are growing across the slope of this image. These are time-lapse photographs from the summertime near the equator on Mars. These are dark streaks that only appear in the summer when temperatures are getting warmer. And some scientists believe, it's quite controversial, that these streaks actually represent water that is coming up from the subsurface when temperatures get warmer. A more recent and really exciting result, or discovery, came just 2 weeks ago, where there was some information that suggested that there's a huge subsurface lake that's covered with an ice cap. So this is I think it's 12-mile long lake with about 1 mile of ice covering it, but there's evidence of a liquid water lake inside of there, and this is based on radar. Here on Earth, we know that such subsurface ice-covered lakes do host microbial life. This is located in the South Pole region of Mars. This is a really cold place. The only explanation for why there could be liquid water at these temperatures, this is about minus 80 Fahrenheit, would be if the water was really salty to depress that freezing point of the water. So if life were to live in here, it would be really salt tolerant and be able to survive the cold. We have evidence here on Earth that microorganisms are capable of this. The next example I'd like to draw on from, again, our own solar system is Enceladus. Enceladus is a moon of Saturn. I think it's my favorite. It is. It's really, get ready. It's a frozen planetary body. Here you can see the ice. But underneath that ice, which the estimates vary between 1 mile to 37 miles of thickness, is a global ocean. All right, so there's a global ocean. Water's really important. There's a global ocean underneath here. This might be hard to see. At the south pole of Enceladus, there are 4 stripes called the Tiger Stripes. These are 80-mile long fractures or depressions that we know are kind of abnormally warm when we compare them to the surface temperatures of the rest of Enceladus. And this picture down here is an actual photograph of these tiger stripes. And I want to draw your attention to these streaks that are emanating from here. What these streaks are plumes of liquid water from that subsurface ocean that are jetting out into the air. Right? I'm going to let that sink in. There's liquid water, right, on Enceladus, and it's coming up from the surface into the air. This is why it's one of my favorites. Right, global ocean. This is an artist's renditioning. Enceladus has a rocky core. Here are these plumes emitting into the air. And some scientists think that what we're having here is that there's the presence of hydrothermal vents in the core. So analysis of the contents of these plumes, we've had some flybys, suggests that there are small organic molecules in that plume. There's methane in that plume. There's carbon dioxide. There's carbon monoxide, right? These are all compounds that we know on Earth microorganisms can use, but these are also compounds that come up from the subsurface, from hydrothermal vents here on Earth. So on Earth, these hydrothermal vent systems are hotspots of life around them, and then those microorganisms are feeding larger organisms, right? Their entire ecosystems revolving around these hydrothermal vents. Could something similar perhaps exist in a place like Enceladus? Something to think about. In a potential future, pie-in-the-sky mission to a place like Enceladus, drilling through the subsurface ice, like through miles thick ice, might be hard. But because it's erupting into the air, perhaps a future mission doesn't even need to drill. Maybe it could fly through those plumes. It could take a sample. Maybe it could bring it back. Something to think about. Europa. Europa is another moon. This time it's a moon of Jupiter. Again, this is another cold planetary body, and like Enceladus, there's also evidence that there are plumes of water from a subsurface ocean that are coming to the surface. Let me let that sink in. There's 2 places in our solar system where we know of liquid water. That's amazing. Part of Europa has these large kind of fractures and cracks in its surface. Scientists aren't entirely sure how those formed. There's some strong theories that maybe this ice that's floating on top of the ocean is moving and flipping over and turning over time, or has in the past. But there's some reddish deposits here. And those reddish deposits are most likely salts. And of course, here on Earth, we know of microorganisms that can survive high salts, right? So one of those examples would be the Great Salt Flats in Utah, where microorganisms are so abundant living on these salts that they actually cause a change in color. These microorganisms are called halobacteria. They're pink in color, and so they make the salt flats look pink. And finally, I'm going to draw on one other example from within our own solar system. There's tons of places I could talk about, but I think Titan is really interesting. And Titan is a moon of Saturn. And Titan has this really thick, hazy atmosphere. So think of LA on like the smoggiest day, and then multiply that by 100, and that's probably akin to Titan. And so this is a photograph of Titan. And a couple years ago, we sent a probe through that haze. And it was a one-way trip. But while this probe was going through the haze, it was sending back photos back to Earth. And we got some really valuable information about Titan. So this is a picture of Titan underneath that haze. And I hope you can see it. It looks kind of familiar. It looks a little Earth-like, right? And Titan actually has lakes, and it rains. There are lakes on Titan. This is an actual photograph that's being false colored, but it is a photograph of Titan, same as here. And these are lakes, but they're not lakes of water. And it doesn't rain water. Temperatures here are really cold. It's about minus 90 degrees Fahrenheit. On Titan, it's raining hydrocarbons. It's raining propane and methane and ethane. So something like gasoline. So these are lakes of gasoline and raining gasoline, right? And so when we think about what we know about life and its need for water, we're thinking about it needing a solvent, right? Something for materials to diffuse through, for things to pass through their membranes. But what about maybe life doesn't need water as a solvent? Maybe life could use another liquid, right? Something like a liquid hydrocarbon. Currently on Earth, we do not know of a single organism that uses anything but water as a solvent. But we can stretch our imaginations to think about habitability in these contexts, and to think about places like Titan in these contexts. And so everywhere I described so far within our own solar system is within the bounds of what we consider habitable, but they're all pretty extreme. And so here I'd like you to think about what are extreme environments to you, right? So what's the coldest temperature that you think life can exist at? Or what's the coldest temperature that you think you can handle? Right? What about, it's different, right? It's a relative term. Some of us love winters in Maine, some of us go to Florida. What about very little to no water, right? How long do you think you could survive without a glass of water? Or very little to no food. The hottest temperature. I think most of us can agree that we can't handle a pool of acid. We couldn't live in a pool of acid. But all of these environments, all of these extreme cases exist on Earth. So here's a picture of the Arctic that's, very cold. Desert systems with very little to no water. Hydrothermal vents that are, spewing hot water all the time. Here's a picture of the Grand Charismatic Spring at Yellowstone. These are as hot, acidic pools. These are environments that we couldn't survive in and definitely could not thrive in, but microorganisms are quite happy in each of these environments. And so thinking about what we know about life, one of the things that we can draw on is that, life exists in some of the toughest places. Life exists in these extreme environments, to these extreme environments. And I'm going to close this section just thinking about some of the things we know about life on Earth. And again, I'm a microbiologist. I think microbes are the coolest. And here are some reasons why. Number one, most of the diversity that we have on Earth is microscopic. We can't see it. And when I use the word diversity, I mean the most amount of different types of species, right? The most amount of different types of species are microorganisms. We need microorganisms for our survival. There's not a single person in this room that would be living if we didn't have microorganisms. Half of the air we're breathing in right now came from microorganisms. The most abundant life form on Earth are microorganisms. But I think really one of the coolest things about microorganisms is that the diversity and how they've adapted to fit every single niche, every single one of these extreme environments is really a testament to how adaptable and tenacious life is and can really form the basis of thinking about life on other planets. So I'm going to take some questions before we go on a break, in which I strongly encourage you to go get a beverage while you think about this discussion question. So please talk with your neighbor over the next 10-minute break, and what would it mean to you if we were to find life elsewhere? So please take a 10-minute break, grab a beverage. Hopefully that'll help the words flow while you talk to your neighbor about this question.
Are we taking questions first?
Yeah, let's take questions, yeah. Okay. I'm going to go back to life on Earth. There we go. Okay, so we've seen this slide. I've already tried to sell you on this idea that microorganisms are the coolest organisms on Earth because they can survive and because they can thrive in some of these really extreme environments. I'm a microbial ecologist. I've spent most of my career trying to look at which microorganisms live in some extreme environments, how are they doing it? And today I'm going to talk about two specific environments. One of them is going to be in the Antarctic. That's work I carried out during my PhD before I got to Bigelow. And then I'll follow that up with some of the work that I'm doing now at Bigelow. And this is a bit more of a work in progress, but I think it's a very compelling story, especially in the context of astrobiology. So this first story takes place in the McMurdo Dry Valleys of Antarctica. And when we think about the Antarctic, most people think about a continent that looks like this, right? A lot of ice, a lot of snow. But there's a special region of the Antarctic called the Dry Valleys. This is a satellite image of it. I hope you can see that there's some brown patches. These brown patches don't have any snow on top of them. And this is what it looks like on the ground. This is the second driest place on Earth. It's a desert ecosystem. And I mean, this could be Chile. The only thing that really indicates that it might be the Antarctic is this very cold researcher that's on a hike in a winter coat appropriate for Maine. And I'm going to read to you an entry from one of the very first explorers of this region of the Antarctic. Sir Robert Falcon Scott in 1905 wrote, "We have seen no living thing, not even a moss. It is certainly the valley of the dead." And for a long time, this notion of this being a dead, barren place was really prevailing. There's no plant life in this area. Birds don't fly into the area. But you know, we now know that life does exist in this environment. What life could that be? Right? Because microbes are the coolest, right? Microbes, there's microbial life in this environment. We know this because we can look at it with our microscopes. We can also extract DNA. All life that we know of has DNA. We can take our soils, extract all of the DNA out of it, and try to identify microorganisms. And I spent about 5 or 6 years of my life studying one place in the Dry Valleys called University Valley, Antarctica. It's a very small valley, has a small glacier at the head of it that's slowly losing mass. It's such a dry place that the water in the ice is constantly evaporating away. This is a shrinking glacier. And as a microbiologist, knowing what I know about microorganisms and how hardy they are, I looked at this environment and I wanted to know what microorganisms live there. And I really hypothesized that some of the most cold adapted and hardiest microorganisms would live in this valley. And so I'm gonna ask for some audience participation again. Given everything that I've already talked about, where in this valley would you look for microbial life? Literally anywhere, under the ground, under the glacier. I think I heard rocks there too. Great. I looked in all of those places, and I'm gonna distill the results of that. First off is I looked in the soil. And one thing that really surprised me was that in the soil there is very little biomass. And by biomass, I mean the amount of cells, the amount of microbial cells that I could find in these soils. So what I was finding was about 1,000 cells per gram of soil that I was looking at. Now, to compare that, if I were to go to, also extreme environments in the Arctic, I would find like 10 million at the very least in Arctic soils in Canada, in permafrost soils. So this was really surprising right off the bat. Then I used a bunch of, the classical suites of tools that a microbiologist used. And I'm going to just give you the punchline instead of showing you a bunch of figures, which is that after 5 years of working on this particular site, I couldn't grow anything from these soils. I could not grow anything in a petri dish in the lab. I couldn't measure any breathing from the soils. So in the same way that we eat a hamburger, we eat food, and we assimilate some of that carbon into our biomass. We also respire out some of that carbon as carbon dioxide. Many microorganisms do the same thing, and so I can use sensitive equipment to measure that carbon dioxide coming off of the soils. I could not measure that in the field at all. I also couldn't measure any available food. I couldn't really find any organic carbon. These are really nutrient-poor soils. And I tried this in the lab at sub-zero temperatures. This is a place that's always cold. Temperatures, even in the summertime, never go above freezing, and I could not get anything. But do you think it's dead? No. When I took them in the lab and I warmed these samples up, performed all of the same experiments, but warmed them up, so now these are temperatures that they don't see in situ. They never see these in the field. The temperatures are never above freezing. What I found is all of a sudden I could grow things and I could measure breathing. And so what this indicates to me as a microbiologist is not that this site is dead, but that these microorganisms that I can see with my microscope are actually dormant. So dormancy is a state that's kind of like a bear in hibernation. They're kind of to grow. One of the ways that microorganisms can do this, and we saw this with our samples, is with a protective spore coat. So they kind of have a hard outer layer, they shrink themselves down, and they kind of bunker down until conditions get good again. And we saw this in our University Valley soil samples. But does this mean that University Valley is dead? Could there maybe be extreme life surviving here? And the answer is yes, yes. And it exists in the rocks. And so the rocks are a really interesting environment in this valley. Again, this is experiencing the same cold temperatures, but kind of like a driveway on a hot summer day. You know, the sun is warming up that rock, so it's a little bit warmer than the atmospheric air. When we crack open these rocks, we find that there's life hidden within it, just beneath the surface. So if you were to walk by a rock, it looks very normal. If you cracked it open, if you were lucky, you would see some of these colorations. This is a bit better of a picture of that. So there's life hidden in the rocks. You can see that there's a lot of sand grain, or like kind of grains in this rock. This is a sandstone rock. What this does is it provides a lot of pore space. So even though this is a desert environment, any available humidity in the air has an opportunity to be trapped within those pore spaces. So these rocks are a little bit warmer, a little bit wetter than the surrounding environment. This is very different than the permafrost environment. We see a green band here. These are photosynthetic microorganisms. So just like plants are photosynthetic microorganisms that use energy from the sun and can, breathe in carbon dioxide and fix that carbon dioxide into organic molecules, these microorganisms are doing the exact same thing. They're taking energy from the sun and they're making carbon into their biomass and also excreting some of that carbon into the rock. But you will absolutely never see a rock that only has a green band in this environment. Never happens. You always see it associated with this black band here. This black band is a group of melanized fungi. Melanin is a pigment. I have it in my skin. It acts as a protectant from the sun. And what these fungi are doing are protecting these photosynthetic microorganisms from harmful UV radiation. The ozone is a bit thinner at the poles. These microorganisms are stuck in the rock. They can't really go anywhere. And so what you have here is a fungal system or fungal community that's protecting the photosynthetic organisms, but the fungi are also never found alone because those fungi need a carbon source, which is exactly what this photosynthetic organism is providing. Here are some microscopic images of these organisms. That's the black fungi and the green organisms. And so what's one of the lessons that we can learn about life from this environment? Well, one is that, sometimes a strategy to deal with extreme environments is dormancy. Maybe sometimes that's just the strategy, it's survival. Another is that, sometimes it takes a community. Microorganisms are actually very rarely found alone in nature. It's a really rare condition. And often the food of one is actually the waste of another microorganism. And these communities are thriving in this environment, but they're not quite possible alone. And so my work here in the Antarctic and trying to measure activity, I was really concerned about questions of activity and how we do it in these extreme environments, are really what brought me to Bigelow and thinking about extreme life. And so when we think about where life exists on Earth, we usually think about land, right? That's probably the image when you think of life. But of course, most of our planet is actually water, and we have tons of ocean, and that ocean is teeming with life. And so what can we learn about the limits of life from the ocean and from the extreme habitats that exist within the ocean? The ocean is, of course, not only water. There's some really interesting environments. Hydrothermal vent systems are one of them that we've heard at some of the past Cafe Size. These are sites that are venting hot fluids and sometimes nutrients and energy sources for microorganisms from the subsurface of the Earth. And today I'm going to be talking about ocean crust, this hard rock at the bottom of the ocean, the very bottom, underneath the muck. And one thing that's really interesting in terms of thinking about this environment is, again, going back to this idea of energy. In the past, in that rock microbial community that I just talked about, the basis for that community was really a photosynthetic microorganism that was using energy from the sun to feed the community. But this is the bottom of the ocean. There is no sun at the bottom of the ocean. It's actually quite far away from microorganisms that are using photosynthesis to provide nutrients. So how are microorganisms in ocean crust living? What are those adaptations? What allows them to survive there? And so the question is, what microorganisms live in the crust and how are they doing it? And so in October, I was lucky enough to get to go on a research cruise with Beth and her group to a site called North Pond. North Pond is in the Mid-Atlantic Ridge, which is an underwater mountain range in the middle of the Atlantic Ocean. And it's a really interesting environment. So let's talk about some of the parallels between this particular environment and the other one. One is that this is also a low biomass system. There's not that many microorganisms that live here. So this is, again, about 1,000 to 10,000 cells per gram of rock, of microorganisms that are living in there. One thing that's a little bit different than the permafrost system is, so think about permanently frozen ground. There's not a lot of influx of nutrients or ways for things to leave. Ocean crust is a little bit different. Has anyone here seen volcanic rock before? Yeah? It's porous, right? It has holes. And so just like aquifers here, for anyone who's had to dig a well, there's water that moves through that rock. And so these microorganisms, even though they may not be abundant, and even though there might be not a lot of nutrients, maybe there's a supply of nutrients that's coming through. Maybe that's one of the things to consider about what makes this environment habitable. So at this particular site, there are some observatories to study life in the crust that have been built. So these are some holes called corks. They kind of think about a cork plugging a hole. And these observatories have some purpose. So they're hole in the crust, and they have some perforations that allow the water that's naturally moving through the crust to flow through them. And this was an opportunity 6 years ago by Beth's team to do a 6-year experiment to look for what kind of microbial life lives on the rocks. And so 6 years ago, some researchers put different types of rock chips in the subsurface in these observatories, and they've been sitting there for 6 years, allowing crustal fluid to flow over them. And the question becomes things like, do different microorganisms like different types of rock? Do different types of rocks select for different microbial communities? What communities are there? What adaptations do they have to live off of those rocks? Can they use those rocks as energy sources? I'm going to discuss some of our preliminary results, but I'm going to pause just to show a few slides of how we got those samples. Because I think that this research is actually kind of difficult to pull off. They're really ambitious endeavors, and in my mind, they're almost akin to going to other planets to get samples. So one is you can't just go down to the bottom of the ocean and get your sample. So they use remotely operated vehicles. So we use Jason. Jason's a submersible. It has two little robot arms. And Jason it's an unmanned robot. It goes over the boat. And I'm showing this picture because I think this really speaks to the interdisciplinarity of the field of astrobiology and indeed of the field of microbiology, because not a single person in these images is actually a scientist. Right? These are pictures of engineers and people that are on ship crews. And so I think this really speaks to the amount of and different people that contribute to getting this type of work done. On the ship, this is kind of what the scene is. So Jason has several cameras around it, and we can sit and look at what Jason is seeing, and we can sample from those observatories. Again, this is an observatory that's drilled into the crust, so our view is only of the surface, so the bottom of the seafloor, but not what's underneath. And to give you a sense of scale, I'm going to show you a brief video. So this is a video of... This is a video of one of these observatories. So for scale, most people here have seen a milk crate before. These are actually pretty big. And so Jason is kind of just doing a sample right now But underneath the seafloor, what you're not seeing are those rock experiments. And so those rock experiments get pulled up by Jason, brought on ship where a bunch of scientists eagerly and quickly work with them. Here's a picture of some of these rocks. They look a lot different than those sterile rocks I was showing earlier. Here they kind of look a little bit rusty, or like they've been oxidized in the subsurface. And again, the questions that as a microbial ecologist that we're asking with these rocks is what microorganisms are growing on them? How much microorganisms are growing on them? And in particular, what I was interested in was this question of, are they active? Are these just microorganisms that happen to be there, or are these microorganisms actually doing anything in this ecosystem? And doing this was something that I really wanted to come to Bigelow specifically to do, and to use the single-cell genomics center here. And this was to use really sensitive single-cell techniques here to measure microbial activity, one single cell at a time. So this is the sensitivity to measure activity in a single microbial cell, instead of trying to measure something like respiration or breathing from all of the cells at once. And the premise behind this is actually pretty simple. You take your sample, your environmental sample, and I feed it something akin to this, right? This kind of looks like something you would eat. You'd probably be fine eating it, but it's not quite food. I use a very similar technique. So I feed my microorganisms a compound that is an amino acid. It's very similar in structure, but it's not exactly the same. And any microorganisms that are active, that are making new proteins in its body, will take up that amino acid and incorporate it into its body. But that fake amino acid is really similar to this donut. It doesn't hurt the microorganism, but it causes them to glow. And so here we have an example of, two of our active buddies and then another buddy that happened to be there. And so from here, we can use a technique called flow cytometry, where we have our active microorganisms, and we can do something called fluorescence-activated cell sorting with the cell sorting facilities here. And so what we're doing is using some specialized equipment that suspends a single bacterial cell or archaeal cell or fungal cell, a single microorganism, in a droplet of water. That droplet of water travels through the instrument and gets binged by a laser. And the way that laser light reflects off of that cell gets detected by the instrument, and from there we can tell whether or not the cell is fluorescent or not. We can also get information about the cell size, but most importantly, we can tell, is it active or not? And if we find out that it's active, we can sort it, and we can put it in a specific pile of, here are the active cells, here are all of the other cells. And from there, I can take out the DNA, I can sequence it, and I can look at the genes within that microorganism to try to deduce what adaptations at the genetic level this microorganism has that allows it to survive in this environment. So what can it eat? Maybe it has genes for motility that allows it to swim towards a food source when food is scarce. These are the types of things that I'm looking for in the genes. And so, like I said, this is a work in progress, but I'll show you some of the results so far. So the first is microbes in the crust are active. So here's some microscopy. You can see some of these glowing cells. These are some of these cells that have taken up that fake amino acid, and now I've caused them to glow. So we know that oceanic crust is an environment where cells are not only just present, like in University Valley, but they're actually active. This is huge. This is great. This is a lot better for me to work on than the Antarctic was. But also something that's really interesting is that we're finding microorganisms that no one has ever cultured before. So I'm going to show the bar graph. Don't be too intimidated by it. There are 4 different rock types here. So this first one is olivine. These are 2. The second one is pyrrhotite, and then there's 2 different types of basalts. You don't need to read any of these words. I just want to call to attention 3 things. One is that the different rocks have different microbial communities. That's number 1. Each of these groups on the left-hand side, those are the cells that I sorted that were active, that were glowing. And on the right-hand side are the total community. This is when I didn't sort based on fluorescence. I just said, here's all of the cells. And so the second thing I want you to notice from this figure is that the active cells are different than the total community. This is really interesting to me when thinking about microbial ecology, because normally what I would have done is just extract all of the DNA from here and try to make deductions about what's happening based on all of the DNA. But now I can be a bit more precise and look at only the active microorganisms and look at what their adaptations are compared to the whole community. I'll get a more accurate picture. And the third thing I want you to notice are all of these words that you may not be able to read in the back that have stars next to them. And all of these phyla are phyla of microorganisms that no one has ever been able to culture in the lab before. So we know they exist because in various environments on Earth, sometimes extreme environments, we can detect their DNA, but no one to date has been able to culture them in the lab. So these are some of the results we're seeing. And you'll have to stay tuned for what adaptations we're finding in these samples. Maybe it'll be a cafe sign next year, who knows? But I'm gonna wrap up this section by, again, kind of talking about what we've learned about life on Earth and how this applies to again, thinking outwards. So studying life on Earth, what does it teach us? One of the things we know is that Earth and the life on it has co-evolved over time. It's happening today, and that evolution, that adaptation has allowed life to diversify and occupy almost every niche that we can think of, the most extreme environments. There seems to be no limit to the potential diversity that we see on Earth so far. And there's an inherent strength in that diversity, right? In some of these extreme environments, it takes a community. Life is adaptable. Life is tenacious. In any environment, life figures out a way to survive and to get energy and sometimes even thrive. Studying life on Earth also teaches us that, if life can thrive in these extreme environments here on Earth, it's very possible that it can thrive on similar environments on other planets and moons. And so I want to close this talk today with this image. Who here has seen this image before, by show of hands? Not that many. Oh, this is wonderful. Great. So this is the photograph. And some of you may or may not be able to see this fraction of a pixel. Here it is blown up to help you out. And this is a photograph of Earth. This is us. This photograph was taken with the Voyager mission. This was taken in 1990. The Voyager mission was supposed to, leave Earth and leave our solar system. And as it was getting to the outer reaches, a very smart person called up NASA and asked them to turn the probe around and take a picture. And they took pictures of all of the planets in our solar system, kind of like a family portrait. And this was our portrait. And the first time I saw this photograph, this photograph honestly had a very profound impact on my life. And in particular, this quote that I'm really happy to share with you, since so many people haven't seen this photograph before. So that person was Carl Sagan, who of course would be able to call NASA, right? So here's what Carl Sagan had to say about this particular photograph. Look again at that dot. That's here. That's home. That's us. On it, everyone you love, everyone you know, everyone you ever heard of, every human being who ever was, lived out their lives. The aggregate of our joy and suffering, thousands of confident religions, ideologies, and economic doctrines, every hunter and forager, every hero and coward, every creator and destroyer of civilization, every king and peasant, every young couple in love, every mother and father, hopeful child, inventor and explorer, every teacher of morals, every corrupt politician, Every superstar, every supreme leader, every saint and sinner in the history of our species lived there on a mote of dust suspended in a sunbeam. All right. And so when we're thinking about life, on earth, we're thinking about this is the story of us. Thanks. I'll take questions.